going to talk about EEG and action potential in brain. EEG is a neuroimaging technique that measures the electrical impulses sent through the brain. It is a non-invasive way to measure the amount of electrical activity that can be detected on the head. To understand how EEG works, we need to know some basic electrical physiology of the human brain. So there are about 10 billion to 100 billion neuron cells in human brain, and majority of them are pyramidal neuron cell. So what we are interested is how messages it pass through the brain. So I draw a diagram to illustrate that. So a secret agent in my diagram, secret agent A pass through a message to B, and B pass through a message to C, and C pass through the same message to D, and so on and so on. Imagine this message is action potential, and those secret agents are neurons. So what we want to know is how do they process, how do they hand over the message. So this is what our brain look like. So when the action potential run all the way down to the axon, once it hit the end, the synapse will be activated and release neurotransmitter. And the neurotransmitter will diffuse toward postsynapse due to simple diffusion. Once the neurotransmitter binds to the receptor on postsynapse, it will induce a postsynaptic potential. It can be either inhibitory or excitatory. What we want to know about is the excitatory postsynaptic potential, which I'm going to call it ESPS. Is the potential that can depolarize the membrane. So compared to action potential, EPSP is very small. However, unlike action potential, is not all or nothing. It can be combined. So once we get enough EPSP, we can trigger action potential onto the next axon, and the message can be passed through. So why do we use EPSP but not action potentials? Because action potential occur at a rate of over 200 Hz, and they are highly localized because they are generated by a single neuron. Therefore, it is impossible to pick up those signals by electrons placed on the scalp. As I said before, EPSP is very small. Therefore, to generate a scalp measurements, we need about 100,000 neurons to be activated at the same time. Once they activate, this provides a strong field, and this field can be modeled as a single equilibrium dipolar current source with some orientation. In our left hand side, we can see this the nerve cell once they get activated, and on our right hand side is how we model it. The use of EEG is to diagnose epilepsy, brain tumor, and accidental damage to the brain. Clinically, EEG is used to monitor the electrical activity in the brain during sleep and during seizure. In order to apply the electron scalp caps are most commonly used. It is made of frame which holds the necessary electrons in place. Also, a special glue is used. The most common electron placement system is 10 to 20 system, in which 21 electrons are placed at intervals of 10% and 20% of the scalp. In our diagram, it's exactly 10 to 20%, 10 to 20 system. The letters F, T, C, P, and O stand for frontal, temporal, central, parietal, and occipital lobes. The number represents the hemisphere position. The right hemisphere is represented by even number, and the left represented by odd number. A typical EEG test takes about 30 minutes and is conducted in a very quiet environment away from interference. The process of applying the electrons takes about 10 minutes. After this, the patient is asked to close their eye and relax for about 10 minutes to produce a reading when they are relaxed. Another reading is then done with their eyes open for about 5 minutes. After this standard routine, what the patient will be given is some different stimulation. One of them is visual stimulation, where soft flashes of light are produced in front of patient eyes. The other one is hyperventilation or sleep deprivation. All of them can trigger a seizure. So here are some normal EEG signals. We come in four different ways: waveform, alpha, beta, theta, and delta. And there's some abnormal EEG features. It's either spike and wave or sharp wave. In our left hand side is a normal EEG traces. We can see all the is mainly consist of different waveform, which is alpha, delta, theta. But on our right hand side one is a generalized epilepsy. We can see we get a lot of spikes and sharp wave in here. These sharp waves should be discrete event that can be separated from background activity. Also, there are two types of seizures, generalized one and a localized. Generalized one seizure will affect the whole brain, while the localized seizure will only involve in one loop of context. In this diagram, on the right hand side one is a generalized epilepsy. So we get some video just for interest. The first video is about epilepsy, and the other one is about seizure. They provide a lot of information in terms of biology and physiology. And for a general interest, if you want to know how EEG can be put on in the children, there's about it. So here's our reference.